I am Dr. Suraiya Saleem, working in Chennai, India. Today I shall be talking about understanding neuron death and drug delivery in Alzheimer's disease. The human brain is a complex organ structured for accomplishing robust physiological behavior and emotional responses. It is made up of numerous neurons and glial cells which interact among themselves to maintain a strong connectivity and integrity. An aging brain is characterized by immense neuron loss and neurodegeneration. Several death modalities are active and responsible for this loss of neurons. Of these, necrosis, autophagy, and apoptosis play pivotal roles in death of neurons. The general mechanisms which govern these phenomena mutually is still not understood. In the ensuing slides, we shall discuss about these in greater details and help create a better understanding of the neuronal death modalities. Neurodegenerative diseases may be defined as a group of complex neurological disorders comprising of loss of neurons either in the peripheral nervous system or the central nervous system. Yet, however, finding a precise definition for neurodegeneration is hard to find. Etymologically, the word neurodegeneration comprises of the words neuro and degeneration. Neuro refers to nerve cells and degeneration may refer to a process in which there is a loss of structure or function. Pathologically, it refers to a condition affecting neurons. Hence, neurodegeneration is an umbrella term which pertains to the progressive loss of structure or function of neurons, or may also indicate death of neurons. Neurodegeneration is commonly used to represent a variety of neurological disorders with heterogeneity in their clinical and pathological symptoms. They also affect various specific population of neurons in specific anatomical functional regions. The specific cause for neurodegeneration is largely unknown, but once its onset is established, it progresses in a largely relentless manner. Pathological conditions which are excluded from the umbrella term of neurodegenerative diseases are neoplasm, edema, hemorrhage. These disorders basically do not involve the primary neuronal regions and hence do not qualify as neurodegenerative diseases. Conditions in which neurons die due to hypoxia, infections, or metabolic perturbations are also not included under the term of neurodegenerative diseases. Neurodegeneration is a condition which accounts for death of neurons in abundance due to several reasons. Neurons are post-mitotic and terminally differentiated. Loss of it exacerbates the problems of neurodegenerative diseases, debilitating the condition of the brain. Studies on the human brain, animal models, and cell culture models have revealed the occurrence of several death paradigms in neurodegeneration. It is therefore clear that multiple mechanisms of cell death govern the devastation caused in neurodegeneration. Crosstalk between these processes is also an indispensable mode of death of neurons. Cell death mechanisms are induced during development for proper organization of cells with respect to space and content, or during disease condition to eliminate unhealthy dying cells. Different neurodegenerative diseases may be governed by different specific dominant death pathways. However, other death paradigms also play minor roles in a death scenario. Different death mechanisms may be acting in varied portions of the same neuron in response to the same stress stimuli. Apoptotic death of neurons has been extensively studied. Acute and chronic neurodegeneration is associated with both apoptotic and necrotic cell death. These death paradigms may be distinctly varied in some situations, while in others they may occur in synchronization. The factors that decide the extent of mutualism between the death pathways are the basic energy content of the cell and the strength of the stimulus. Studies showed that the spatial location of neurons governed partly their fate when subjected to ischemia brain injury. The neurons at the periphery underwent apoptosis, which was rescued by caspase inhibition, while the core neurons could not be rescued by inhibition of caspases, thus indicating that they died through necrosis. When the cells are unable to combat the stress, which is beyond their tolerance level, they undergo necrosis, an acute non-apoptotic form of cell death. The mechanism of cell death are conserved from nematodes to humans. Pathological neuronal deaths may occur by necrosis in the brain in neurodegeneration. Necrosis in neurons is triggered by excessive energy depletion. Loss of energy leads to collapse of ionic gradients which are required for sustenance of neurons, which is followed by a buildup of glutamate at the synaptic clefts. Increased accumulation of glutamate at the synaptic clefts causes necrotic death of downstream synaptic target neurons. A common anomaly of neurodegeneration is metabolic perturbation, due to which there is excessive release of reactive oxygen species, which leads to necrosis. Proteolytic systems also largely influence necrosis. Release of cathepsins D and E have been noted to cause necrotic death in transient forebrain ischemia. Spatial localization of cathepsins play critical roles in neurodegeneration. Young rat brain cortices exhibit lysosomal localization of cathepsins, while cytosolic localization of cathepsins is observed in aged rat brain cortices. Accumulating evidence reveals that instead of assuming a chaotic architecture, necrotic cell death may be following a well-organized pattern. Extensive studies on the probable molecular mechanism of necrosis brings forth only a limited number of key players and their mechanism that could be involved in the process. The autophagic response has been reported in various pathophysiological states like neurodegeneration, cancer, and cardiovascular diseases. Studies have shown that autophagy plays a role in the removal of misfolded proteins that accumulate within the cell. Whereas autophagy is needed for physiological conditioning, non-dividing cells like neurons are notably sensitive to changes in autophagic degradation. As autophagy is important for the clearance of aggregate-prone proteins, the thought of autophagy failure as a mechanism predisposing to death has relevance to pathological processes in a variety of diseases. Defects in autophagy would lead to accumulation of broken organelles and degradation of durable proteins and aggregates, leading to their accumulation within the cell. Under normal conditions, Autophagy is a gift at basal levels to maintain proper physiological conditions. The demand for cellular internal control through autophagy is especially vital in post-mitotic cells like neurons and myocytes, and dysfunctional autophagy has been related to cell death in various neurodegenerative diseases. Apoptosis mediates the precise and programmed natural death of neurons and is physiologically a necessary process in maturation of the central nervous system. However, premature apoptosis or an impaired apoptosis regulation is involved in pathologic processes of neurodegenerative diseases and results in varied chronic illness states like Alzheimer's, Parkinson's, Huntington's, amyotrophic lateral sclerosis, spinal muscular atrophy, and diabetic neurological disorders. Activation of neuronal cytomembrane receptors might end in caspase activation, increased calcium levels, and generation of reactive oxygen species, and contribute considerably to programmed cell death. High calcium levels and ROS are known to contribute to the mitochondrial pathway of apoptosis. Mitochondrial pathway is initiated by a range of signals as well as the BCL2 proteins, high calcium levels and ROS. These signals accumulate at the mitochondria, leading to the discharge of many pro-death molecules into the cytoplasm. Mitochondria, additionally to their role in energy production, contribute to cell death by releasing pro-death molecules into the neuronal cytoplasm. One pro-death molecule with specific implication in neurodegenerative diseases is cytochrome C that exists unremarkably within the electron transport chain on the mitochondrial membrane. Cytochrome C associates with a apoptosome complex with procaspase E nucleotide and APOF1. This complex then works as a cell death signal by promoting proteinase activation. The relationship between autophagy and apoptosis is quite complicated. In some instances, autophagy proves to be a protective mechanism, while in other cases it seems to induce apoptosis and lead to death of cells. This differential functionality of autophagy seems to be dependent on circumstances. Cell death orchestrates life during development and also delivers the essence of life. Alzheimer's disease is a progressive neurodegenerative disease which accounts for about 80% of dementia among the elderly population worldwide. Clinically, Alzheimer's disease is characterized by the gradual impairment of cognitive functions, which ultimately culminates in death of the individual. Specific regions of the brain are affected in AD. The specific neuropathological sites in AD include the hippocampus, amygdala, the temporal and frontal cortex. With an increase in life expectancy as the population ages, there is an increase in the number of patients suffering with dementia. According to the World Alzheimer's Report 2018, 47 million people are living with dementia worldwide, and this number will increase up to 32 million by 2050. The report also provides the information that even in most of the high-income countries, people with dementia either do not have access to or are very poor to access appropriate healthcare awareness. The scenario in these high-income countries is such that only 50% of people suffering from dementia receive diagnosis, whereas in low and middle-income countries, the number of people being diagnosed drops to only 10%. About 110 years ago, in 1906, a German psychiatrist and neuroanatomist, Alois Alzheimer, reported a peculiar, severe disease process of the cerebral cortex to the 37th meeting of Southwest German psychiatrists in Tübingen. In his report, he described the history of a patient, a 50-year-old woman, Mrs. Auguste Dieter. On performing autopsy of the brain, he observed extensive atrophy of the cerebral cortex. Thin slices of the brain tissue were stained with silver salts and examined. Alzheimer's disease being a multifactorial disease caused by complex interactions between genetic, epigenetic, and environmental components. The early onset of autosomal dominant form of AD that occurs at an age earlier than 60 years can be caused by three gene mutations: amyloid precursor protein, preselinin 1 and 2. These mutations alter the processing of APP, resulting in abnormal production of amyloid beta peptides. Late onset AD may be caused due to environmental factors or mutations in ApoE. Early onset AD sets in patients of the age group 30 to 60 years of age. This is a rare form of Alzheimer's disease. It represents only 5% of the total AD patients worldwide. Usually, the cause of early onset AD is not known, while it is noticed that such cases are mostly inherited from the previous generations. This type of AD is referred to as FAD, or the familial Alzheimer's disease. Single gene mutations are mainly the cause for FAD. They may occur in any of the chromosomes 21, 14, and 1. The result of each of these mutations is the formation of abnormal proteins. Abnormal APP is a result of mutation in chromosome 21. Mutation in chromosome 14 and chromosome 1 leads to the production of abnormal presenilin 2 and 1 respectively. Late onset AD, the most prevalent form of AD is the LODE, which occurs in patients of over 60 years of age. The exact pathological reason behind the occurrence of this late onset AD is still not clear. However, reports suggest a variety of causes behind its occurrence, which include genetic, environmental lifestyle factors, or a combination of all. These play roles in the disease susceptibility of the patients. As mentioned earlier, single gene mutations are responsible for the early onset AD. They do not seem to have any role in the load. However, a single genetic factor, apolipoprotein E, ApoE, resides on chromosome 19 and is suggested to play a pivotal role in increasing the disease vulnerability among its bearers. The main function of ApoE protein is to help in the transport of cholesterol and fats in the bloodstream. In the CNS, ApoE is mainly produced by the astrocytes. These proteins help in the transport of cholesterol to the neurons via the ApoE receptors. The three most frequently observed alleles of ApoE are ApoE2, ApoE3, ApoE4. ApoE2 is less prevalent and may play a role in protection against the disease. Patients that develop Alzheimer's disease due to a mutation in ApoE2 do so at a stage later than patients with mutations in ApoE4 would have developed the disease. ApoE3 is said to be a neutral protein which does not play any role in disease development or progression. ApoE4 mutation is the most predominant form of mutation found in almost 40% of patients with late onset AD. An improvement in human health and an increase in longevity have been the results of biomedical researches that have been done through ages. However, Unfortunately, we have seen an increase in the age-related disorders, which largely include neurodegenerative diseases. The symptoms of these disorders mainly include impaired memory and cognition, defects in movements and control, aggressive behavior, and emotional imbalances, ultimately leading to death. Alzheimer's disease, as previously described, is one of the most common forms of dementia among the elderly. A huge range of animal models have been used to understand the underlying biochemical mechanism in Alzheimer's disease. Animal models that have been used to facilitate understanding of AD include worms, fishes, flies, sea urchins, ascidians, rodents, and higher mammals. Moreover, the use of simple model systems like cell culture models have also been extensively used to obtain key revelations regarding AD diagnosis and therapeutic strategies. We have used the PC12 or the pheochromocytoma cell obtained from the rat adrenal medulla to study Alzheimer's disease. This cell line has several advantages like easy pharmacological manipulation, easy maintenance and culture, and highly specialized differentiation. Having an embryological origin within the neuroblastic cell line, they can easily differentiate into neuron-like cells. They share several neuron-like properties when differentiated with the nerve growth factor. They acquire similar neuronal morphology, they release neurotransmitters by vesicles, they show long neuronal processes with vesicles. In our lab, we study Alzheimer's using PC12 cells by treating them with either amyloid beta oligomers or depriving them of NGF by giving them anti-NGF to simulate neurodegenerative conditions. So these two act as the stressors for creating the model. PC12 cells have been used extensively for studies on hypoxia, exocytosis, and prion diseases. Cell lines have played critical roles in enhancing scientific knowledge and temper. Further advancement on cell line research is the use of primary cultures. In our lab, we work with primary cultures of cortical and hippocampal neurons. The cortical neurons are isolated from the 18-day embryonic rat brains. Once plated, the cells are maintained in DMEM F12 medium, supplemented with insulin, glucose, transferrin, progesterone, putrescine, and selenium. Cultured neurons are subjected to treatment after a period of 6 days. The primary hippocampal neurons were cultured from the 18-day embryonic rat hippocampus. Cells are plated on PDL-coated plates cultured in neurobasal medium, supplemented with B27, and maintained for 3 weeks. After 3 weeks, the cells are used for treatment. We checked the activation levels of death modalities like autophagy and apoptosis in our AD models. We found that both these death paradigms were active in AD. LC3 or the light chain protein 3 and P62 are well-known autophagy markers. LC3 remains in the soluble form in cells. After the induction of the first gene in autophagy, that is ULK1, there occurs a schematic upregulation of a number of autophagic genes which help in the double membrane structure, which is an autophagosome formation. The structure can be identified as an autophagosome only after the addition of LC3 on its membrane. When there is an induction of autophagy, then LC3 is cleaved into LC31 and LC32. The cleaved form is further conjugated to a phosphatidyl ethanolamine group. This lipidated form of LC3 binds to the membrane of the autophagosomes and therefore is used as a marker for autophagy. The LC32 remains on the outer wall of the autophagosomes. Presence of LC32 marked membranes indicate the occurrence of autophagosomes, which go on to indicate an induction in the process of autophagy. We checked the levels of LC3 in APP-PS1 double transgenic mice by performing immunohistochemical staining of the brain sections from these APP-PS1 transgenic mice and the wild-type mice. We observed there was an increase in the punctated staining of LC3 in the transgenic mice as compared to the wild type. Now coming on to the PC62. P62 is a ubiquitin binding scaffold protein. It co-localizes with the ubiquitinated protein aggregates. P62 contains an N-terminal PB1 domain which helps in the essential formation of polymers of P62. The formation of these polymers occurs when the acidic surface of one P62 PB1 domain binds to the basic surface of the next P62 PB1 domain. The PB1 domain is reported to have a role in the autophagic degradation of P62. It bears a C-terminal UBA ubiquitin-associated domain via which it binds to the ubiquitinated proteins. P62 is also known as sequestosome 1, which is a factor that targets cargo for autophagy. P62 acts as a carrier of unwanted proteins to the autophagosome to enable their degradation. The generalized function of P62 therefore lies in ensuring aggregation and subsequent turnover of the unwanted ubiquitinated proteins. The protein is itself degraded by autophagy, but if at any point of time there occurs an accumulation of P62 within the cells, it points out to a different condition altogether. It has been reported that P62 within the cells is accumulated when autophagic degradation is inhibited. Hence, considered as a marker for autophagy flux. We checked the levels of P62 in APP-PS1 transgenic mice and we observed that there was an increase in staining of P62 in the TG mice as compared to the wild type. We further checked the level of apoptosis in the AD model. For this, we checked the propatotic proteins like BIM, PUMA, and performed tunnel assay. We observed an increase in the expression of the apoptotic proteins in our AD model. So these go on to indicate that there is an unupregulated level of apoptosis and autophagy in Alzheimer's disease. We next checked whether our target proteins, that is a TRB3 and Becklin, both implied in ORI autophagy and apoptosis, were required for evoking neuronal death in response to amyloid beta. In order to analyze this, we interfered with their expression by using previously described shRNA constructs. Now, these constructs efficiently blocked the induction of the protein levels in amyloid beta treated condition. Primary cultured cortical and hippocampal neurons and differentiated PC12 cells were transfected with the shRNA construct, and the shRNA used as control was shRAND. Transfected live green cells were monitored and counted under the fluorescence microscope at different time intervals. We observed that downregulation of these genes blocked neurodegeneration as seen from retention of neuronal processes and also led to significant survival of these neurons. BH3-only proteins, that is a BCL2 homology domain 3 of the BCL2 family, are the surveillance of cellular stress, orchestrating cell death via apoptosis in neurons. Both intrinsic as well as extrinsic cell death signals are initiated by the BH3-only proteins of the BCL2 family. The members of the BCL2 family of proteins contain either one or up to four of the BCL2 homology domains. The members are the pro-survival proteins BCL-XL, BCL-W, MCL1, and the pro-apoptotic proteins that is BAX, BAC, BIM, and PUMA. It is through these domains that a complex interplay of interactions of these proteins ultimately determines the fate of the cells. Our studies revealed that knockdown of the pro-apoptotic proteins BIM and PUMA could rescue neurons from death due to overexpression of BMF. It brings to light the crucial role of the BMF in causing neuron death in an AD model. It also highlights the cooperation of the apoptotic proteins in orchestrating the neuron death mechanisms. The zebrafish Venio is emerging as an increasingly successful model for translational research on human neurological disorders. They show a high degree of neurological and behavioral resemblance with that of humans. It is highly validated as a powerful vertebrate model for investigating human neurodegenerative diseases. The neuroanatomic and neurochemical pathways of zebrafish exhibit a profound resemblance with the human brain. Physiological, emotional, and social behavioral pattern similarities between them have also been well established. Interestingly, zebrafish models have been used successfully to simulate the pathology of Alzheimer's disease as well as tauopathies. Their relatively simple nervous system and the optical transparency of the embryos permit real-time neurological imaging. The zebrafish model can be pivotal in bridging the gap between the bench side and the bedside. The zebrafish can be used for genetic manipulations by adopting techniques like viral chemical vectors, CRISPR-Cas9, morpholinos, tilling, and talons. The zebrafish is therefore becoming an increasingly successful model to understand Alzheimer's disease. We in our lab have established an Alzheimer's disease model by using amyloid beta in the zebrafish. So we have used amyloid beta 1 to 42, which is the most highly toxic species observed in the AD patient brain. In order to confirm the presence of amyloid beta in the brain, we performed immunohistochemistry by obtaining slices of the brain sections of the treated versus control brain, and we found that there was positive amyloid beta staining in the treated sections as opposed to the control sections, and this indicated that there was no false positive staining. So we observed the presence of the amyloid beta that was injected in the zebrafish brain. Now, in order to understand the effect of the amyloid beta in the brain, we performed a series of behavioral assays. They included the spatial alternation task, the novel object recognition, the light and dark assay, and many such assays were performed. So, in one of such assays, which is the light and dark assay, in which generally it is observed that the control fish have a tendency to stay in the dark conditions because they inherently like to be in the dark grooves in the pond. But it was found that in this case, upon being injected with amyloid beta, they did not show a proper comprehension of the environment. They seemed rather confused, so they did not spend any considerable time in the dark compartment, and they were moving in an erratic manner. Further, when we performed the novel object recognition assay, we found similar behavior from the amyloid beta treated fish, in which they refused, or they rather had no recognition, no memory of the object that they were habituated with. and they couldn't discriminate between the novel and the previous object, unlike the control fish. So this clearly indicated that the injected amyloid beta had a profound impact upon their memory, their learning, their ability to comprehend, and thus it acts as an important model to understand Alzheimer's disease. Now coming on to drug delivery to the brain. Now, this has always remained a challenge for the research community and the physicians. The blood-brain barrier is physiologically a network of capillaries and cells to protect the brain from the invasion of any foreign particles. The blood-brain barrier is a physiological block to the brain. Reports suggest that the blood-brain barrier is compromised in the disease conditions. The success rate of recent drug delivery systems is less than 10%. It's very poor. This is mainly due to the fact that the existing drug delivery technologies unfortunately lead to several unwanted side effects. The research community has explored various avenues for drug delivery of safe and targeted drugs to the brain. Nanotechnology-based drug delivery is believed to have immense promise in the field of therapeutics. With the advent of nanotechnology, synthesizing structures in the order of a billionth of meters have been proposed to overcome the hurdle of the blood-brain barrier. Nanoparticles are sized between 1 to 100 nanometers, and they help transport the drugs to the brain. Nanoparticles are the objects of choice for delivering drug to the brain because they can not only target the brain specifically, but they can also overcome the hurdle posed by the blood-brain barrier. Nanoparticles can be manipulated according to the requirement of the experiment. Some of the parameters which can be altered or manipulated are stability, size, hydrophobicity. Additionally, these nanoparticles can be administered to the model system in a variety of routes, which include oral, inhalation, injection, or perinatal. Targeting of specific tissues can be achieved by conjugating the nanoparticles with various ligands like antibodies or proteins. The various types of nanoparticles that can be synthesized are liposomes, micelles, dendrimers, lipid or polymer-based nanoparticles. Nanotechnology, therefore, has become an area of extreme interest with the industrial and the research community for delivering drugs to the brain. Nanoparticles with increased efficiency and accuracy for neurospecificity and the ability to overcome the blood-brain barrier are now being developed. Nanoparticles targeted to the brain must be spherical, of less than 100 nanometers in diameter, molecular weight should be less than 400 daltons, positively charged. In our lab, we have established nanoparticles from naturally occurring polymers and have characterized them by using field emission scanning electron microscope, transmission electron microscope, atomic force microscope, and the FTIR. These nanoparticles have a size of less than 100 nanometers in diameter, and they have the ability to cross the blood-brain barrier. Further experiments to study the efficacy of the nanoparticles in an Alzheimer's disease model are underway. To summarize my talk, I would like to emphasize on the fact that in an aging brain, neurons die due to several paradigms. Hence, targeting one pathway is not going to be helpful. Considering Alzheimer's disease, which is a multifactorial disease, a combinatorial therapy is required for successful therapeutic intervention in AD. I hope you have found this talk informative and interesting. I hope you've enjoyed it. Thank you.
Thank you, Dr. Suraiya, for presentation.